Welcome back students for Susi's biology. Hope you have enjoyed and learned the last chapter Animal Kingdom, and within 5 minutes we have finished. And like that today let us revise the important points to remember in the topic cell cycle and cell division, and keep watching, don't miss it, and I assure that this video will be 100% useful for you once again. And for new subscribers I have given the link in the description part, and you can make use of it for the last chapter Animal Kingdom. And let us go on to the topic cell cycle and cell division. Cell. Cell is defined as the structural and functional unit of all living organisms. So it is the basic unit. And the cell theory, what is meant by cell theory? It accounts that all the living organisms are made up of cells and cell products. And there is a epigram, or it is a Latin phrase. Do you know what is omnicellula and ecellula? It is a famous Latin phrase proposed by Rudolf Virchow. In the third image, you can see him. Rudolf Virchow proposed this Latin phrase. It is also called as an epigram. So it means the cells arise from pre-existing cells. And you all know that cells are divided into two types. So prokaryotes and eukaryotes. Based on what? So based on the presence and absence of the membrane-bound nucleus, cells are classified as prokaryotes and eukaryotes. And who proposed the cell theory? So these three peoples, you can see them in the image. So M.J. Schleiden, Theodor Schwann and Rudolf Virchow. So they proposed the cell theory. And here you can see the difference between prokaryotic and eukaryotic cell. So in the image you can view the shape and size of the prokaryotes and eukaryotes. So prokaryote means they do not have a true nucleus, and only 70S type of ribosomes are present here, and whereas in eukaryotic cells 80S ribosomes are present and true nucleus is also present. So you can see the tabular column, the difference between the prokaryotic and eukaryotic cells. Pro means before and eu means true or after. So before carrion and after carrion. Carrion means nucleus. So before that is 3.5 billion years ago prokaryotes are evolved, and 1.5 billion years ago eukaryotes are evolved. And when compared to the size, prokaryotes are very smaller. They are just 1 to 10 micrometer in size, whereas eukaryote it is 100 to 1000 micrometer in size. And prokaryotes are of unicellular type and eukaryotes are multicellular, and all the organelles are absent in prokaryotes whereas they are present in eukaryotes. And the location of genetic information means it is present in the nucleoid region, whereas all the genetic material is present inside the nucleus in eukaryotes. And the DNA, it is found to be circular and it is scattered, and it is not circular in eukaryotes whereas they are circular in prokaryotes. And if you take the reproduction means, in prokaryotes asexual mode of reproduction occurs, whereas in eukaryotes sexual mode of reproduction occurs. And all the prokaryotes, they are anaerobic and eukaryotes are aerobic. So these are some of the simple differences. And let us now see about the cell cycle. So all the cells, they arise from the pre-existing cells, and this occurs by the process called cell division. And there are sequence of events which occurs during the cell division, and these sequence of events through which the cell passes from one division to the other, it is known as cell cycle. This is the first line is the definition for cell cycle. And the human cells, they can complete the cell cycle in 24 hours, whereas the yeast cells, they can complete in 90 minutes. So these are the two important points. And now we can see the cell cycle in detail. So they are divided into two main phases, namely interphase and mitotic phase. Mitotic phase is otherwise called as M phase. So what is meant by interphase? The interphase is otherwise called as resting phase. Here the cell grows in size and the DNA replicates in this interphase. And the interphase is again subdivided into three phases, namely G1, S and G2. Gap 1, synthesis, and gap 2. So these are the three phases, and I have given the important events that occur in each phase. So at G1 phase, the cell grows, the organelles undergo duplication. At S phase, the DNA replication and chromosome duplication occurs. But the chromosome number will remain the same, that is diploid. And G2 phase, it is a period of cytoplasmic growth and protein synthesis occurs here. And there is a phase called G0 phase. So here it is an inactive stage. So heart cells and nerve cells, After G1 phase, they will enter into this inactive stage, and this one is called as quiescent stage or G0 stage. So now you can view in the diagram. So the interphase, all the three phases G1, S and G2 are given here, and mitosis, it is followed by karyokinesis and cytokinesis. So in G1 what happens? The cellular contents are duplicated, and in S phase the DNA replication occurs, and in G2 phase the cell can double check the duplicate chromosome for any errors, and they will make the needed repairs. So this is the cell cycle, and G0 is given here, it is the cell cycle arrest. Now we will come to the mitosis. So the mitosis, it starts with karyokinesis and ends with cytokinesis. And during this mitosis, there is no change in chromosome number in the daughter cells, and hence it is called as equational division. And again this mitosis is divided into four phases, four stages. That is prophase, metaphase, anaphase and telophase. So it is given in the diagram, you can see it. You can remember it as pro, meta, ana, telo. So in the order you can remember. So prophase, metaphase, anaphase and telophase. So what happens in prophase? So just points to remember I have given here. So chromosome condensation occurs. So the chromosome will be condensed, and the movement of centrioles to either poles, that is the centrioles move to the either poles, to the two sides, opposite sides, and there is a disappearance of nucleolus and nuclear membrane, and you can see the thread-like structures, they are called as spindle fibers. So spindle fibers are formed here. That is the very important point to remember in this phase. Spindle formation occurs. And in metaphase, these are the two events, important events. So all the chromosomes, they are aligned at the metaphase plate. So the center portion, like equator, it is called as the metaphase plate. And the spindle fibers attached to the kinetochore of the chromosomes in the metaphase. So all the spindle fibers, they get attached to the kinetochore of the chromosome. The central part of the chromosome is known as kinetochore. So they get attached to the kinetochore. And during anaphase, the centromeres will split, and they result in daughter chromatids, and the daughter chromatids, they start to move to the opposite poles. So you can view in the diagram. So all the daughter chromatids, they move to the opposite poles. So this happens in the anaphase. And telophase is the last phase, and the chromosomes reach the opposite poles, and the reappearance of the nuclear envelope, nucleolus and cytoplasmic organelles occur. You can see the new envelope formed here. So they reappear, and the mitosis restores the nucleocytoplasmic ratio. That is the ratio between the nucleus and cytoplasm. So it is restored here, and this telophase involves in cell repair and replacement. And the next one is meiosis. So these are the important points about meiosis. So the meiosis is very essential for the gamete formation, and the specialized diploid germ cells, they undergo meiosis and they result in haploid daughter cells. And the chromosome number gets reduced. So this meiosis is known as reduction division, because the chromosome number gets reduced to half in the daughter cell. So they are known as reduction division. And the meiosis occurs in two stages, namely meiosis 1 and meiosis 2. So meiosis 1 means it is the reduction division, and it has four stages, namely prophase 1, metaphase 1, anaphase 1 and telophase 1. And similarly meiosis 2 is also followed by four stages, namely prophase 2, metaphase 2, anaphase 2 and telophase 2. And in between meiosis 1 and 2, there is one more stage that is called as interkinesis. That is the stage seen between two meiotic divisions, interkinesis. So you can see all the stages of meiosis 1 and 2 given here. So the interphase is followed by prophase 1 and metaphase 1, anaphase 1 and telophase 1 in meiosis 1. And prophase 1, what happens? Spindle formation occurs, and in metaphase at the center part all the chromosomes arranged and aligned together, and then they will separate to the two poles, and in telophase they get separated into two daughter cells. Again meiosis 2, it is followed by four stages, namely prophase 2, metaphase 2, anaphase 2 and telophase 2, and then it will enter into the cytokinesis process. And most of the events that function to differentiate the meiosis from mitosis occur in prophase 1. And this prophase 1 is divided into 5 distinctive substages, and they are known as leptotene, zygotene, pachytene, diplotin, and diakinesis. So let us see what will happen at each stage. So in leptotene, the chromosomes begin to condense. So the chromosome begins to condense and they are attached to the nuclear membrane through their telomeres. So condensation of chromosome occurs in the leptotene stage. So you have to remember the each stage and what will happen at each stage. That is very important. What will happen at leptotene? The chromosomes will condense. So condensation of chromosomes will occur in leptotene. And the second stage is zygotene. So here the synapses begins with a synaptonemal complex and it forms between the homologous chromosome. So formation of a synaptonemal complex. So synapses between homologous chromosomes. This is the important event occurs in zygotene. And in pachytene, crossing over of genetic material occurs between the non-sister chromatids. So pachytene means crossing over stage, crossing over between non-sister chromatids, and diplotin means synapses ends with the disappearance. There is a disappearance of the synaptonemal complex and the homologous pairs remain attached at chiasmata. So dissolution of the synaptonemal complex is the important point here in diplotin. And at the stage of diakinesis, there is a dissolution of nuclear membrane and the chromosomes become full condensed and the nuclear membrane disintegrates. So prior to metaphase 1. So these are the stages of prophase 1. So hope you can understand easily about the cell cycle and cell division. Thank you for watching and keep on watching. Kindly share and comment your views and subscribe for the next chapters. Thank you students.